given to Professor Alexandra Konradi. She will share the major results from Almazov National Research Centre within a framework of activities of world-class research center for personalized medicine. Professor Konradi.
Yeah, so dear Chairman, dear members of the advisory board, do you hear me and see my slides well?
Yes, perfect.
Yes, I have a very difficult task today because I'm obliged to present data from 27 research projects and about 200 different researchers who are working inside these projects. Believe me, it was not very easy to select the slides which to present today, and it will be a very quick overview and a summary of what we are doing during this year. So just to remember you, especially to those participants who are for the first time at our advisory board meeting, this is the mission of our world-class research center, which was designed to 2020. And its major goal was to create a biomedical R&D ecosystem in St. Petersburg, especially, which can develop and what is very important, implement in clinical practice the technologies based on personalized medicine, including very well known to you assessments of genetic risks, pharmacogenetics, disease modeling, technologies of genome editing, and others, just to implement this in clinical practice. As you can remember, all the studies in this world-class research center is grouped in 4 major directions. The first 3 are mainly performed in Almazov Center, and the last one concerning infections and antimicrobial therapy is mostly designed inside our partner, the Institute of Experimental Medicine, which will be presented by Professor Dmitriev. However, we also have some studies in infections. Let me show you the general groups of projects which can reflect the 3 levels of personalized medicine implementation as we see it for the future development of our center. So the first group was the infrastructure for personalized medicine, because you cannot develop the specific innovations, drugs or something new if you do not have a good infrastructure and methodology. So we were aimed to create different cell models, animal models, including gene editing and creating new models of genetic diseases. We created an infrastructure for bioinformatics and created different new even algorithms to be implemented in genetic research. And also what was very important is to do several platforms for drug development and drug testing, just to be inside the program. The second group of projects were projects aimed at creation several new treatment options for our patients and to change the protocols which can be ready to be included in the guidelines. And we also performed a lot of clinical studies and planning to perform them. These are, first of all, personalized treatment approaches based on different biomarkers. Sometimes there are new indications and algorithms to treat several diseases, especially oncology, and also risk scales and personalized prediction models for different diseases. And the last one, but possibly the most important part of our work, is to create the real innovations. And we will tell you about the vaccines, drugs, and CAR T cell products, several medical devices, and a lot of IT projects based on decision support system, mobile applications, and several other ideas. Just in brief, what we have now in numbers, just for you to understand what we are speaking about. For this year, we have from this center 42 publications in Q12, general 87 publications in Scopul, and 16 on revision. We have 12 patents, so that's in innovations, 3 drugs and also radiopharmacy, 2 vaccines in projects, 6 mobile applications and disease support system, which can be in future implemented in the real world practice. And we have now a very big staff. It's about 220 leading researchers from 33 PhD students and more than 40 different labs and departments. So starting from the first group of projects, it's populational genetics and gen-environmental interactions going to non-communicable diseases. Just to describe what we are doing, so we have several cohorts of patients coming from the population sample cohorts, some unique biobank data from Leningrad siege survivors and their offsprings, obesity and diabetes cohort, gestational diabetes, acute coronary syndrome, and several others. What we do for these cohorts is genotyping, and we performed the first GWAS analysis made in Russia for the whole genome sequences and also the next generation sequences, both the targeted and the whole genome. For these cohorts, we are performing a very precise phenotyping. First of all, cardiovascular, then metabolic phenotyping, coagulations. We also go to electronic medical records of these patients in the hospital and collect different data from visualization laboratory and so on. And we also have data about nutrition and exercise from specific questionnaires. And then we follow up this cohort of patients, calculating mortality, cardiovascular events, especially recurrent events, development of diabetes medicine, change in target organ damage, and many others. Just in brief, this is the published study, which was a proof of concept for polygenic risk score calculation protocol for Russian populations. So we performed genome-wide association study, and with genotype imputation, we created a polygenic risk scores for different phenotypes in Russian population. And on the right, you can see the comparison of our data with the famous cohorts from UK biobank and also from Japan. And you can see that the Russian cohort is very close to European one and rather far from, for example, Eastern one, which we genotyped here. Now we are inventing the new methodology, which was introduced in genetic research recently in 2021. And so we try to test here the local protocol for the so-called ultra-low coverage whole genome sequencing, which can help us to use it instead of genome-wide association studies, and we can improve individual inherited risk prediction for these patients. Now the 30 DNA samples from St. Petersburg were compared by microarray genotyping and also this protocol of ultra-low coverage, and we performed the genome imputation and have generated the sample data specific to Almazov Center protocol. Going to gene-environmental interactions, very briefly, we showed and recently published a study where we showed that patients with concordance between inherited risk and lifestyle risk factors, they can demonstrate, for example, supernormal vascular aging. At the same time, only genetic risk is not enough to predict, for example, target organ damage and vascular aging by phenotyping. Another study was concentrated on the model predicting sustainability of metabolic health in obesity individuals, and it is valid in 68% of cases. A very interesting study is now under publication for the transgenerational effects of the exposure to famine, because we are following not only the survivors of the Leningrad siege, but their offsprings for the first and second generation. And we showed that they have some changes in dietary pattern and, for example, renal damage. And this publication is under review. From the practical point of view, in this part, we created a very elegant system. It is a mobile application. It has 2 versions for iOS and Android. And it can predict postprandial blood glucose for female patients who are pregnant and have diabetes, especially gestational diabetes. And this complication can help our patients avoid hyperglycemia. And now we are in a clinical study, in a randomized clinical study for this their companion implementation, and preliminary results revealed that there is a decreased postprandial glucose level in women who are using this application in general life. We have a very big cohort of obese patients and are creating the personalized treatment, and we tried to evaluate predictors of the weight reduction response to different agents. So here you can see the list of general predictors of the good response to weight lowering therapy, specialized predictors for GLP one receptor agonists, and also for reducin, where we found a lot of negative predictors, including insulin resistance and hypertension and advanced age. And the last project in this part is the acute coronary syndrome project, which was done specially during this year. We managed to collect 500 patients with acute coronary syndrome hospitalized to Almazov Center, and to collect a lot of biosampling on the 6 hours after angiography, 3 days and 3 months. And we do the mass spectrometry analysis for different metabolic predictors for the recurrent events. For example, on these slides, you can see the list of fatty acids and also ceramides who are under evaluation as the predictor of the recurrent events in this cohort of patients. Coming to the rare disease and undiagnosed disease, on this slide you can see the summary of our concept and research architecture. I will start from the end and say that all this research was aimed to design an excellence center for the undiagnosed and rare diseases, both in children and in adults. And we work in major clinical areas as cardiomyopathies, autoimmunity and autoinflammation, sleep disturbances, neuromuscular diseases, monogenic rare diseases, several congenital diseases, and others. So what we need to establish such a center? The first, the major disadvantage of rare disease is that they are rare. So it's difficult to collect a big sample and to collect enough data to have a very convincing conclusions. So we do the data accumulation. For this purpose, we created the rare disease registry and a specialized cardiomyopathy registry. We also perform an artificial intelligence algorithm for genetic disease detection on the populational level, because many of these diseases are underdiagnosed in the general life, or lately diagnosed when it is too late to administrate any treatment for this. So we organized a biobanking and genetic data storage, which is what is also very important. And we also do, together with our partner, a specialized genetic program to store the data and analyze genetic data quickly compared with the phenotyping. And later on, we can do a lot of genetic testing, especially NGS, of course, for these cohorts of patients. We confirm our findings from the possible mutations by functional studies, creating cell models, for example, iPS cells. Then we creating animal models on fish and rodents, knockout models, and several other. And these can lead to special targets and even possibly for gene therapy. And we have one project closer to for creating a gene therapy drug. Starting from the registry, here you can see the brief description of the registry of unknown rare and genetically determined diseases. Now 75 diseases are in the registry. You can see the list of them. And how it is organized. So the registry is collecting genotype and phenotype and the follow-up of this patient. And we collect both somatic outcome and neurocognitive outcome for the quality of life of our patients and also the data from the family. And of course, this can help to create the evidence-based model and also the value-based model of treatment for these patients, including cost optimization. So you can see here some examples of the data that we have in our registry and the follow-up of these patients. Especially for this project, our genetic lab, led by Anna Kostereva, created different panels for the targeted NGS sequencing. So the most frequently used is the cardiac panel, then arrhythmogenic syndromes, titin and titin-associated genes, and hypertrophic cardiomyopathy. Also, a very interesting panel is for ciliopathies and congenital structural defects, congenital diseases of endocrine system, and many others, tumors of endocrine system. So those who work in NGS understand that design of any panel is a very precise intellectual work just to do it perfectly. Here you can see one example of how we use the genetic basis for example, congenital diseases, to see the new targets and to understand if it is possible to understand molecular mechanisms of very difficult to analyze in diseases. And on the left part of this slide, you see the targeted gene panel sequencing and the genetic variants, rare genetic variants that were revealed in our lab. And on the right side, you see the functional molecular studies to reveal the functional part of these changes. So we took the nasal brush biopsies and we analyzed by high-speed video microscopy the cellular beat frequencies and their pattern, which is changing in these genetic defects. Then RNA transcript analysis and other functional ideas just to determine how in these rare genetic variants is behaving the targeted cells which are involved in this pathology. Here is also an example of our big research in cardiomyopathies and especially laminopathies with the muscular phenotype. And you can see how we go from experimental cell models to therapeutic solutions. And on this slide, we can see that we created transgenic lines of mice myoblasts who are expressing the pathogenic variant, which is associated with lipodystrophy and muscular alterations. And in these skeletal muscular diseases, we can also see the involvement of myocardium. And on the right side is the single cell RNA transcriptome analysis, where you can see how it is functionally work and expressed in different cell types. So the next part is animal modeling. This is also a rather complicated slide, but in brief, we are working to understand molecular and cellular mechanisms of neurogenesis and neurodegenerative and even psychiatric disorders using genetic editing technology. So we created in Dania radio a mutant model with the defect of OLIC2 gene, and we showed that it has a significant deficiency of the spinal cord motor neurons, also posterior root ganglia, sympathetic and enteric neurons, as well as the several disorganization of adrenal chromaffin cells. So, and these results are very important to understand the mechanisms underlying the development of these neurodegenerative diseases. The next part of our neurodegenerative projects is analyzing the function of the cells from the neural crest. because these cells, they can be considered as a new cellular material for regenerative medicine, and the regenerative medicine and cell therapy is also a very important part of the personalized medicine. So we created by our molecular studies a unique atlas of gene expression of neural crest cells, and also their followers, so the cells that are developing from these cells, and we can see that they can develop to Schwann progenitor cells and have similar transcription profiles, and so they are able to specialize into different cell types as glia, neurons, neuroendocrine cells, and many others. And this opens for us the future therapeutic potential. Going to autoinflammation and autoimmunity diseases, here you can see one project which was aimed to rather rare autoinflammatory disease. It is Still syndrome in adults, Still syndrome. And we created the atlas of clusters of different cells based on analysis of 13 samples. And you can see a lot of cells that were phenotyped. And we built an expression profile for this part of patients. And we can follow these patients during the therapy and specialized immune therapy and see how this pattern is changing during treatment and to predict the response to treatment. Going to the more clinical project, one example of the work again in Still syndrome, it was a novel therapy algorithm which was proposed by other investigators and published in a rheumatology journal recently, where we proposed to treat these patients together with colchicine, rather safer and very cheap drug, together with non-steroid anti-inflammatory drugs, with a very good response, and 65% of cases they have significant improvement in clinical manifestation. And only after the failure of this therapy, the patients are going to biologic drugs who are much more dangerous and expensive. Now, what about very interesting projects on excessive calcification? As far as you know, not only in cardiology, but also in several other fields, the pathological calcification is becoming a problem, and it is growing up like an epidemic of the 21st century. So we created a special testing model to test the effects of different chemical compounds and genetically engineered constructions to suppress this pathological calcification. And we found a new substance, crenicacetate, which was completely suppressing the cell differentiation of the aortic valve to the calcification process. This is not a new substance. It is approved to treat cancer, and it is effective to prevent calcification at the same doses that is effective to treat some cancer patients. So we hope that if we have a success in the preclinical and clinical studies, we can have a new indication, possibly registration for this drug, which is already adopted as an in oncology. And the last project in this field is familial hypercholesterolemia. So you know that there is still some discrepancies between clinical and genetic diagnostic scores to detect these specialized patients of a very high cardiovascular risk. So we created a clinical registry for these patients with different phenotypes, performed for them precise imaging for subclinical atherosclerosis, genetic testing for all known and even unknown genes that can be involved in the system, and also proteomic analysis. And in progress, there is also the animal models for the CATP zebrafish, its knockout model, and also the rodent model for PCSK9 defect, and also for the LDL receptor gene. Going to gene editing therapy, we selected a very rare disease. It is really very rare, and it is X-linked inheritance. It is called BART syndrome. These children do not live very long. They have changes in immunity, neutropenia, infections, and also cardiomyopathy. And the survival rate is very low. So it is associated with so-called TAS gen. So here you can see that we created a special line, knockout cell line, with the knockout of the TAS gen and also C12 gen. And you see that in this model, the protein synthesis is inhibited. We tested functionally these cells and have shown that oxygen consumption is reduced. So we see that these cells with knockout cells are genetically are not very energetically effective. And now we are using not only the adeno-associated constructions, but also lentivirus construction for cloning, lentivirus plasmid for TAS cloning, and selecting the specific target to create a drug that can treat this very life-threatening pathology. So the last big project in Almazov Center is, of course, oncology, including oncohematology. Here you can see the major tumors in which we are working. This is central nervous system, neuroendocrine tumors, of course, oncohematology, which is a classical part of the Almazov Center, but the tumors of pancreas, cervical cancer, breast cancer, and several others. We develop diagnostic tools, including radionuclide techniques for PET-CT, and also some artificial intelligence system for analyzing visualization. What about treatment? Of course, the major idea is to have new mutation and new targets, but you understand that it is not very easy. We do new CAR T cell design, and then I'll show you an example. We also create antibodies, especially bifunctional antibodies, which is a new step in the development of the targeted therapy in oncohematology. We do develop new indications for personalized prescriptions in oncology patients, also create anti-tumor bacteria and viruses, which will be presented in the next presentation, some new devices, and we also work in personalized rehabilitation and treatment of therapy complications, especially in cardiac oncology. Here is the design of the new radiopharmaceutical, which is targeted to diagnose the metastasis and of any phenotypes of prostate cancer by PET-CT. And it is assigned not only to PCMA, but also for GRPR. And during this year, we established the optimal conditions for the synthesis of the project, selected effective precursors, validated procedure for manufacturing, did the quality standard, and together with the Uppsala University, we did the animal studies for efficacy and subchronic toxicity, biodistribution, the dosing and diagnostic efficacy of this compound. And next year, it will be involved in the real clinical practice. In going to artificial intelligence diagnostics, it is the brief description of the project of the screening of cervical cancer based on coloscopic screening. And we created the network algorithm, which is capable to distinguish between benign and malignant cervical lesions. And in this year, during this year, we have an accuracy of the model about 92%, and it can distinguish normal vision and pathology very quickly and very effective. And the very similar project in vascular, in endoscopy ultrasound. You can understand that doing artificial intelligence with the ultrasonography is much more difficult than compared to CT, MRI, or any other high-resolution images. So there is a big noise and different artifacts when you work with the ultrasound imaging, but at the same time, we collected a very big database, more than 1,000 images for different tumors of pancreas. And now the primary test of algorithm gives us the accuracy of 85%, which is very promising for the possible use of the system in the screening of ultrasound. And going to molecular profiling of the central nervous system, just in brief, we try to create the precision cancer therapy in several central nervous system. And here are the data from medulloblastoma in kids. As you know, of course, the profiling of medulloblastoma in adults is very well described, and you can find the images together with the genetic phenotyping in the specific databases. But going to the kids, it is not so well organized, and these data from any child are rather new, and also the therapy for the kids is not so very well established, because the clinical trials in kids oncology is not a very easy to organize, and usually our kids they have prescriptions of the adult drugs which are administered off label for them. So we found, we made a genotyping of several tumors. We found new missense and nonsense mutations in these patients. And of course, when we will collect a lot of this data, and we are aimed to have more than 100 patients by the end of our project, we can speak about new prognostic factor and the possible target to treat this patient. The same is for neuroendocrine tumors. These are not very frequent disease, and our center is working in neuroendocrine tumors for many years and collecting patients. So what we did now, we did the microRNA analysis for 32 pituitary adenoma samples, and you can see more than 500 unique microRNAs, RNA molecules that were detected in the tumor expression profile. Some of them were associated with the invasive development of the tumor, and some of them on the right, they were associated with the recurrent tumors, which were with the recurrence after surgery treatment. So this also can serve as a possible future marker to select the technique of surgery and possibly some indications for new therapy. Going to oncohematology, our big project is in CAR T cells. And here you can see the design of bifunctional CAR T cells, which can be a much more effective therapy and subsequent control of hematology disease as the monospecific CARs. Because these bispecific CAR C cells, both to CD19 and CD20, they can effectively lese the tumor cells, even in the absence of one of the markers. And on the right side, you can see that they are more effective in vivo analysis just to suppress the tumor activity. This was absolutely newly designed. Our center worked a lot in oncohematology, especially in myeloproliferative neoplasia. And we worked together with our German partners on resistance of target therapy of this neoplasia. So you can see that after some period of time, so these patients can become treatment resistant, and this is a big problem for oncohematology. So there are some intracellular mechanisms and extracellular mechanisms which can be responsible for this treatment resistance. So we did a lot of studies about the dysregulation and storage-operated calcium entry in myelofibrosis, also the role of desaminases in chronic myeloid leukemia, and the possibility for immune escape induced by myeloid-derived suppressor cells in chronic myeloid leukemia. And all three of these projects are now in progress, and we hope to find some approaches to overcome this treatment resistance, for example, by inhibiting desaminases and inhibiting these myeloid suppressor cells. Another project in oncology is both a diagnostic and treatment project, is the use of the fact that heat shock protein 70 is expressed on the tumor cells. So we developed a special peptide, which is called RAS70 peptide, which can target this heat shock protein 70 positive tumors, for example, brain tumors. And if we combine this agent together with immunofluorescent agent, we can see that it is we can match and indicate where and differentiate the tumor tissue from the healthy tissue during surgery operation. And for our so we hope that this can help to better differentiation during surgery for the volume of the tissue which should be removed, and according to calculation, it can double the survival rate of the patients with severe malignant brain tumors. And the same approach we are going to test to palliative local treatment of end-stage and complicated pancreatic tumors, because heat shock protein can also be the substance for injection of intratumoral agents, just to treat these, unfortunately, palliatively treat these tumors in the pancreas. And on the right side, you'll see a small device that we are developing together with our industrial partner. It's a plastic universal biliary stent for treatment of obstructive diseases. It is also a palliative treatment, but it is improving the quality and length of life of these patients. Together with the Institute of Radiology in Moscow, we develop a new anti-angiogenic and hypoxia-oriented anti-tumor agent. And you can see on this picture some experimental studies on metastatic carcinoma S37 in mice. And you can see that this compound is inhibiting the development of the tumor after tumor transplantation, and it is a possible compound to be used in oncology. Of course, it is only under preclinical development. And the last two slides in oncology is about rehabilitation. And here you can see the work of our group of the working with post-mastectomy syndrome. And they develop personalized programs for rehabilitation. And here you can see that they found a lot of markers of the development of this syndrome. You know that it is very important problem all over the world, even in Russia, about 1 million females, they do experience such a problem during their life. And here are the markers of fibrosis, the damage of the central nervous system, and the markers also of peripheral nervous system damage, who can serve that this syndrome can appear in the future. And another part is the cardio-oncology, and here is the data of the group working with checkpoint inhibitors therapy. We in cardiology understand that this is a great challenge for cardiology, so development of new oncology drugs is leading to a huge amount of patients with some cardio-oncology problems and cardiotoxicity. And our group revealed several predictors and negative changes after 3 months of follow-up of these patients, including decrease in ejection fractions and also increase in several humoral markers. And another part of this project is the problem of venous thromboembolic complications. You know that thromboemboli is one of the leading causes of death of all oncology patients, and tumors of the central nervous system is not an exclusion. So for these patients, it's a very dangerous complication. And we included 75 patients into the prospective study, and created a specific scale who can calculate the risk of venous thromboembolism for these patients, and even do a decision support system for the doctors just to calculate the risk and to personally change the treatment to prevent these complications. And by the end of my speech, just a few words about infections. Of course, in 2020, we came across a new challenge. It's COVID-19. It was not included in the first version of the program of our center, but we could not escape for not doing the research in it. And so we included several projects. During these years, we have about 4,000 patients hospitalized here in the clinic of Almazov Center. We performed for them standardized examination and treatment, autopsy, the biobanking, and prepared the data for the genotyping during the exit phase. Now we are doing the follow-up of these patients, including risk calculation also in hospital and after hospital mortality. And we are planning to have the 3-year perspective or possibly even more. What we also register is different events: cardiovascular, neurology and cognitive function, depression and anxiety, diabetes, peripheral thrombosis, and many others which are registered in our database. So I'm sorry. So here you can see the mortality risk calculator, which was tested in our center, and now it is tested for the work in other hospitals who are still dealing with COVID-19 patients. Fortunately, we do not have a lot now in Russia, very few patients, but we still have some severe cases. And during this period, we have 4,000 patients and 470 deaths, and 11 factors by artificial intelligence were included in the model from the 46 significant, and the model predicts, unfortunately, the lethal outcome very precisely. We worked a lot in the possibility of the impact of vitamin D level and COVID-19 severity. And we showed that in reality, the deficiency of vitamin D was a risk factor for having severe COVID-19, but we did not show any treatment effect of administration of vitamin D in COVID-19 patients. We collect a lot of data, and here you can see the SARS-CoV-2 viruses in the myocardium, in pancreas, and even in the brain. These are autopsy findings of our patients, and this is also under the further investigation. This year we performed the pilot genotyping for the COVID-19 patients for 380 patients, and you can see on the right these data are under construction. So we found more than 4 million DNA variants, and you can see that we found one variant associated with the infection of COVID-19, which was not found in other populations. And in future, we will try to see if it is really has any functional meaning. We also follow the metabolic status of our patients and showed that that there is a very high prevalence of impaired glucose metabolism in post-COVID patients, and many of them have diabetes and impaired and pre-diabetes, quite 1/2 of patients, and this can be due to some alterations of the pancreas and also the consequences of the administration of the high doses of steroids during treatment. This is also a challenge, and in general we showed that after discharge, and very successful discharge from the hospital, those patients who have any cardiovascular complications, so the 15% of them are readmitted during the 6 months of follow-up, and 38% of them they have cardiovascular diseases, and 31 urgency of cardiovascular diseases. So we follow these patients and see how it is influenced in the future by this infection. These are our major publications in this field. And coming to the end, a few words about education, because World-Class Research Center is not only a research center, but it's an educational center. We developed 3 new programs in 2020, for the 2 for master's degree and 1 for PhD in clinical medicine in oncology, and we have new admitted students for them this year. And here are also the data about new programs for physicians and postgraduate education for both in genetics, in fundamental oncology, and many others who are performed on our platform. And we are very happy that we have a lot of young investigators who are interested in these projects and attending them. So thank you for your attention.
Thank you very much, Professor Konradi. Well, according to the agenda, questions and discussion are meant to be well organized after the next presentation. But since some of the board members are obliged to leave probably prematurely, so probably are there any questions to Professor Konradi? A couple of questions probably, and then we will proceed to the next issue.
Well, if I may pose a question. Dr.
Yes, sure.
This was extremely impressive, and thank you very much for this overview. I just wonder how you create a balance between funding these various activities. How do you do that? Do you fund according to publication impact factors, or according to number of PhDs, or number of external money support? How do you do this?
Yes, thank you for this very challenging question. So we at first we organized it as we have the whole funding. It is divided into these 4 projects, and the money which are going to specialized section, they are fixed. So just inside the leading scientists, they can keep some balance between projects. But during the first years of our work, we did not organize any penalties or some ideas for those who do not have some outstanding results or publications, because we have some projects that are based on a big background, and we have some projects which are very new, and the people start from beginning with nothing behind. And so we decided to help them to grow up and to do something really impressive. So just even now, it is the time just to select and to understand what is really working and what is promising for the future. And this is also one of the tasks of advisory board to have the, if I am right, to express their opinion, what they do like better, and to have the advice possibly which should be done more precisely, or this is not so interesting for the future. This is one of the idea of organizing this board. And next time on our next board meeting, we will share with you the plans for the future, for the development, and just also to understand where are we going. It is not very easy for us, because the situation now that is that the government expect from these research centers even more that they can do. There's a lot of things, and especially quick innovations and quick products. And you can imagine that several things cannot be done so fast, but we do our best with our team.
Thank you.
Thank you, Professor Konradi. And Professor Michel Kamajda.
Yes, good afternoon, dear colleagues. So I would like first to congratulate Professor Konradi for this very impressive summary of a number of different projects. So it's really amazing. So Alexandra, I have 3 questions to you. One is on the registry on offsprings of survivors of the Leningrad siege in the 1940s. You said that there was evidence of renal damage in these patients. What is the underlying cause? Do you think it's an epigenetic cause? Could you please comment? Second question is, you said that you have designed an algorithm based on artificial intelligence to identify genetic disorders in the general population. Could you tell us a little bit more? And third and last question, what is the rationale for testing colchicine in Still's syndrome? Thank you very much.
Thank you. Answering to your first question, Michelle. I'll better start from artificial intelligence, for the first question. So we cannot explain the direct mechanisms because our molecular study are not done, all of them still now, and we did not perform for them yet epigenetics, for example, some targeted or the whole changes in methylation or several other things. But in literature, it is described that there are some transgenerational effects for those people who were exposed to severe environmental factors, and their grandmothers and their parents, they were exposed to a very severe famine lasting for about 2 years. And I do not know any other population where you have so long restriction of calories. So we think that this is epigenetics, but we cannot explain now, especially possibly there is the reduction of the nephron number, but we cannot prove it now, but we are just thinking how to explain it. Possibly it's just an accident, so nothing. So we need to look further, and we are continuing to collect this data. But what is interesting that they have the same eating behavior as their parents. Yes, but possibly this is of course due to family behavior. Yes, and going to artificial intelligence. It was not a very complicated program. What we did, we gave to our industrial partner several markers of what can be, what should be screened. So, for example, for Fabry disease and several other diseases, what compliance, what signs in electronic medical records can be suspicious for having this disease, for example, in cardiology departments, in children's and many others. And then they created a network algorithm and tested it on our database of more than 60,000 pediatric patients. And surprisingly, we were really surprised. It immediately found the 7 patients with Fabry disease according to the science, so it's really working. Well, now what we are going to do is to test this algorithm in other hospitals on their electronic databases. So the idea is to take the whole electronic medical records from the hospital, taking them to this algorithm and having a selection, so the list of patients who can be suspicious. And then you examine them and check if the algorithm is right or not. So we think that this implementation, it can increase the number of patients who are diagnosed in the correct time with such diseases as Fabry in several times. But we will see how it will work in practice. But
excuse me to interrupt you. Is it based mainly on clinical, electrocardiographic, imaging, or biological data?
Both, everything what can be important, but not in all patients. We have all of them. We have a lot of missing results. So it's working on electronic medical records. It's just cross-sectional idea. Yes.
Michelle, may say a few words. The main idea in this artificial intelligence to screen classical electronic medical records in typical hospital, in typical children's hospital, trying to find in them patient with some rare diseases.
Thank you.
And the last question, Michelle, I'm sorry, I did not catch it exactly.
It was, why did you test colchicine in Still syndrome? What is the rationale for testing colchicine?
So the rationale was that colchicine is widely used for idiopathic pericarditis in patients. And in Still syndrome, pericarditis is very frequently observed. So the idea was that the idiopathic recurrent pericarditis is like underdiagnosed Still syndrome, possibly, and their treatment can be the same. And so we tested this treatment and have a very good response. And rheumatologists from Europe are very interested in this idea because it's cheap and effective.
Sure. Thank you very much.
Thank you.